We're doing well on time, and I'm going to launch into this talk. This is a new attempt for me, so again, I would like some audience feedback on this. I haven't prepared anything quite like this before for a patient symposium. But what I'm hopeful to accomplish is to tell you some stories of how some of the very important advances in medical oncology have gone from a laboratory setting, like where Dr. Holman works, into the clinics where Dr. Kujakar, Dr. Anabaka, and myself get to give these things to patients. So to understand clinical research, you really need to understand what the types of that are, and that's in part where I'm going to start this. I'm going to hit the key steps in drug development and use a very important story in targeted therapy. And then finally, these stories are inspirational to me. This is what got me fired up when I was a scientist myself and in medical school. It's what really drove me into this field. And so I'm hopeful you find it as exciting as I do. So research is expensive. That's the downside. And when you look at the costs in cancer drug development, it goes from big to ginormous. And it does that over a course of several steps. What we do in the lab really can be considered target identification. What makes this cancer tick? Why is it growing faster than all of the other normal cells that are surrounding it? Why does it seem to invade the normal checks and balances that try and keep it in a controlled place? This is the work of multiple scientists around our country, each of them with funding in the order of $1 to $10 million to fund a lab project similar to what Dr. Holman's working on in her mice. So from those targets, we then try and design personal drugs to hit them. That's not so trivial either. You can think of this process as very similar to a kid playing with Legos. That's what I think of the best chemists. And they're in there, they're saying, I'm going to put this orange block here and this green block over here, and it's going to fit perfectly into this little area in my Lego construction. And that's kind of what drug design and synthesis is all about. Well, it doesn't stop there. Once they get a little hit, they got to make that little hit stronger. And so they optimize this, tinkering with that Lego and tinkering with its formulation, i.e., how do I get this safely into a patient? Is it going to need an IV? Can it be taken by pill? Maybe we can deliver it through the skin, or in the case of these newly injectable therapies, let's put them right in the tumor. Well, these are, they sound on the surface to be relatively simple decisions, but to make things able to behave in those different environments requires very careful formulation. And of course, we want to do this in animals. Before any drug should see a human patient, it needs to be proven safe in multiple dozens, if not hundreds, of preclinical studies are done in order to prove that safety before we even get close to giving this to a human patient. What happens here is really a go, no-go decision. And if you're at a pharmaceutical company or you're an investor and you're trying to decide, am I going to take the leap and start trying to give this to human patients, there's almost an exponential leap in cost associated with it. And it's a really important crux, because that compound has to be scaled up. It has to be produced in a way that it can be given to the thousands, if not sometimes millions of patients that need access to the drug. They have to go through a pretty intensive regulatory filing with our Food and Drug Administration. They look at all of this data very carefully before we would ever consider doing what we call a phase 1 trial. And there are 3 phases of clinical research, which I'll outline here briefly with my next slide. But by the time you get to the final phase, an average phase 3 clinical trial, like the one that piloted and defined TVEC as an FDA-approved drug, is a $100 million project. Huge money involved in getting these things into patients. So phase 1 research, what is this? This is primarily about safety. So we've done all that work in the lab. We have a good indication that this seems to work in the cancer we want to study. Now we have to take the leap of faith and find people with a cancer that is the right kind of cancer to match the drug that don't have other therapeutic options. As a patient, this is a scary place to be, and there are people in this room that have participated in phase 1 research. This means that you're getting a medication that has very promising science behind it, but unknown benefit, and even more importantly, unknown safety. And so it is far and away the most experimental of any of the kind of research that we do with human subjects. We do small groups, so these are folks that are going to go in groups of 3. They will receive the new experimental medication, and they're followed very closely, usually for many months, to see whether that group of 3 patients is going to tolerate the drug okay before we expand this to more and more numbers. Well, if it looks good in the first 3, we add 3 more, and we increase the dose slightly. The reason this dose has to come up is we want to get to that level where we're going to maximally hit the cancer. That's the ultimate goal here, is we want to get rid of cancer, every last little bit of it, but we want to make sure that's safe. So we gradually increase the dose in groups of 3 patients until we get to something that we can feel comfortable being safe. That level is called the maximum tolerated dose. That means that, and this is actually 1 level below where we hit the ceiling. We try and create that safety buffer for future studies. This is not a set-in-stone dose. There is actually flexibility in this, and as I go on through the talk, I'll explain how that might be adjusted. But it really is the starting point by which all drugs eventually will make it into clinical practice. The other things that we want to learn in these trials is, how does this drug process through the body? Does it clear through the liver and out through the stool? Does it go through the kidneys? How quickly does it escape the bloodstream? How long are the levels going to be maximally in order to fight the cancer? And these are all of the questions that we're asking here. This requires lots of blood draws. So the patients that have participated in this know that this is a very intensive, very committed thing on the part of the patient to be able to give us the information we need to do this for others. And then finally, we want to know, is this hitting our target? And oftentimes we'll either use blood draws, and your white blood cells that are circulating through the blood are actually a very good surrogate for us to see whether or not it hits the same targets in the cancer. But even better, and this is why melanoma is so important to us from a research perspective, is that we can biopsy the tumors. Those of you that have had tumors underneath the skin have probably had multiple biopsies along the way. Those contributions are like gold in terms of our understanding of the disease biology and how a drug can work. A phase 2 study is something where we've got our safety. Hopefully we've got an early signal. We're seeing some responses and we're getting more excited about the potential for this drug in other patients. But we obviously need to go slow and carefully, and we expand our population. Now we're looking at about a 50-patient trial to do a phase 2. During that phase 2, this is all about efficacy and safety. So now we're trying to get a reliable statistical measure of how well does this drug shrink a tumor. Often we have the same pharmacodynamic and pharmacokinetic studies done along the way, so they're repeated again. And then finally, we want to know, how long can this treatment last? How long will these patients go before their tumors start to grow again? If we're successful with a phase 2, the drug works well, the tumor is shrinking, we then have to go on and conduct something called a phase 3 trial that compares it to the standard of care. This is where it does get kind of thorny, because in melanoma, we have a history of not having a standard of care. Especially in the adjuvant setting where I work, for years we have had a debate over what the exact drug should be, and so many of the adjuvant trials that we conduct are going to have a placebo. Why is a placebo important? Well, particularly in the setting of immune therapy, as many of you know, your immune system is actually very tightly tied to your mood, your belief in the future, your anticipation of good things to come, and all of those things can be impacted by even a placebo. We've studied this for years, and we know that people can actually have a very profound placebo effect. In immunology studies, it is probably one of the most important things that we can design into a trial. Nowadays, we are able to put placebos on top of a known drug, and there's a couple of examples of these clinical trials we'll be talking about in a little bit, where patients are not just getting nothing. They're actually getting the gold standard of therapy, and a new drug like T-VEC is being added on top of that gold standard, and that's an example of where placebos are used. Stratification is a process by which we want to balance those two treatment arms. Well, what does that mean? It really has to do with risk. So when you try and balance two groups of patients, not all melanoma looks the same. There are some patients with relatively small amounts of tumor under their skin. They're young, they're healthy, they have no medical problems. Other patients come with more challenging circumstances, lots of tumor, maybe other medical problems like heart failure. And we want to make sure that the two groups are balanced in terms of those health characteristics and age and tumor biology. So at the end of the trial, we know that the new drug was better. And not just better because we picked all the good patients for one side and all the hard patients for the other side. We, of course, do continued safety monitoring. This is part and parcel to every element of clinical research we conduct. The safety of our patients is really a priority, and sometimes we learn things as those trials expand to large numbers. The 1 in 1,000 events start to occur, and we have a very robust monitoring system that allows reporting directly to the FDA and other site investigators if there is any kind of surprise that happens along the way in clinical research. It gets widely disseminated, and sometimes studies are paused. If a bad thing happens, research stops. We collect our breath. We go back to the drawing board and we make sure it's safe. But at the end of the day, we're hopeful to get a new drug approved, and that, fortunately, has happened many times in melanoma. So I'm going to tell you this story. This was the story that got me excited to go into cancer medicine. It happened in a laboratory down the hall from where I was training, and it was the development of really a first-in-its-class targeted therapy called Gleevec, and I know many have read about this drug. It was a collaboration between industry and academia, so it does require kind of a village to do these things. It required a big pharmaceutical company in addition to several who were working in labs, one a chemist and one a physician scientist. And what they did that was so unique at the time is it was the first example of where they took knowledge of exactly what the target was in their cancer, and they designed a drug to go after it. They designed special tests in the lab, like what Dr. Holman was talking about earlier. This is why you're giving these tumor specimens to place in these mice. It's this step here to test it in a really carefully designed system to prove it before we go to patients. They then picked the right patients to do this in. And then finally, they had to push through obstacles to drug discovery. I'll say a few things about this. You remember my slide a few moments ago that talked about the cost of research. When you go from that early phase to the advanced phase, the hundreds of millions of dollars that it requires to do that is not trivial. And so if we're in cancers like melanoma that are not the most common cancer, it does require someone very passionate about the research to sometimes say, hey, you know, this may not affect a lot of people nationally, but boy, is it an important thing, and it really does affect a lot of important people. It may not be breast cancer or colon cancer in its scope, but boy, melanoma is important and we have to go after it. And this is still a conversation that needs to be had among our industry colleagues, and it is regularly. So what are the numbers of CML? This is a blood cancer. There's only about 5,000 men and women who will be diagnosed in 2010 in the US. That means about 2 people in Utah annually. That's not a very big number. It's about 2/3 the incidence of testis cancer and only about 2.5% of the breast cancer incidence. Melanoma kind of falls somewhere there in between. And what makes it unique is it's introduced by this genetic swapping of BCR and ABL to create what we call a promiscuous kinase. Well, what does that mean? This is that genetic swap. This is chromosome 9 and chromosome 22, and these things have been inappropriately exchanged. This is after birth. This is a genetic accident that happens as cells divide, and it makes this thing called the Philadelphia chromosome. Well, when that happens, again, the kinase that is supposed to be very specific starts talking to everybody. It's like the chatty Cathy of the kinome. This is signaling all of these different targets downstream to divide, 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 grow, grow, grow. Well, obviously, this is not what you want at all from the point of view of cancer biology. So this paper and work led by Brian Drucker was published in 1996. I was a trainee at the time. And the really important point, this is kind of a busy slide, but this became the whole concept behind how we design drugs. And I'm going to try and walk you through it in a way that makes as much sense as possible. Each item here on the left column is a different target within a cancer cell. These are kinases. You can think of them like switches. So if there's like a relay of switches that has to all be turned on in order for a cell to divide, well, these are that parts of that series of switches. In CML, BCR-ABL is the one key switch, and you don't want to hit all the switches, because if you hit all the switches, this drug could be horribly toxic, right? You need these things for normal function, biology. Well, what Brian Drucker and coworkers showed is that they could design a molecule that was super specific. It takes micro amounts of this particular drug to completely shut off the BCR-ABL switch, and yet it takes massive amounts of that same drug to even start touching these off-target things. And that's what makes this true precision medicine. The impact of this has been huge. So in the years prior to Gleevec, the survival time in CML was not good. And by 5 years, 50% of patients would be expected to die of their disease. Nowadays, today, that number is close to 95% survival. And that's my dream for what we can do in melanoma. I believe that this same kind of thing is possible. And in a lot of ways, this is the model by which we do it. Some really exciting math about it is a simple discovery in a very small cancer can have a huge impact in lives saved over time. And I did this sort of back of the math envelope calculation and came up with about 200,000 patients being alive in 2020 as a result of that discovery back in 1996 that otherwise wouldn't be here. So it's for that reason that I think the basic science that we talk about today is so important to being able to get melanoma curves to look like this. And great things happen to the investigators. This is Brian Drucker, and he's carrying the torch for the 2002 Winter Olympics in Salt Lake City. This is actually mid-afternoon in Portland, Oregon, where I grew up. This is a sunny day. But Brian is a great mentor. He worked very closely with my wife. He actually was present when my son was born. And I'll never forget this quote. He came to the room and my wife said to him, "Oh, look, the man has arrived." And he said, "Nope," points to my son, "There's the man." So he's very generous and humble, and he's just a terrific collaborator. So how do we move past Brian Drucker? What are the things that we're doing now at the Huntsman to try and drive this even faster? Well, Sunil Sharma, who leads our phase 1 therapeutics program, has a project here that really is designed to take many of the ideas that we have and put them into supercomputers. And these computers will take that Lego process of molecule design that I told you about and turbo boost it. So they can crank through multiple different steps and trials, and instead of having to screen tens of thousands of compounds to get into a patient, they can screen hundreds of compounds instead. And as you can imagine, this is a factor of 10 or 100 in terms of discovery pace. This is important in melanoma. So BRAF inhibitors were developed with this same kind of concept. They used a computer system. This is a group led by Gideon Bollag and Meenert Herlin. And they drove down to a small number of compounds and found one that hit the BRAF oncogene, which is present in about half of all patients with melanoma. This is Gideon Bollag, and you can see they developed a very similar table to the one I just showed you ago from Brian Drucker's lab. And in that group, they showed that it took almost nothing in terms of their BRAF inhibitor to completely shut off the BRAF switch, and buckets of it would be required to shut off these other important switches that are needed for normal cell health and biology. This is a ribbon diagram, and essentially is what the computer sees. So when it's trying to put together one of these Legos, it's actually modeling it with ribbons and lines and putting the molecule right into the pocket in order for them to get a good hit. The other exciting thing that happened with this molecule in melanoma was the selection of the right patients. This work led by Keith Flaherty, who many of you know. It did take a tribe of clinical investigators here, but Keith really is the kind of key leader who pushed this over the finish line. And this was done in a phase 1 trial. So this is some of the first, these are experiences of some of the first patients receiving this molecule designed on a computer and then in a lab. And what they were able to show is that about 70% of the patients had a response to that single drug. Wow. Off the charts response rate, incredible. And many of the patients on this study went from being so full of cancer that their pets looked like Christmas trees to being completely quiet, sometimes in the span of only 8 weeks. Just jaw-dropping results. Tumors the size of softballs were shrinking dramatically over those 8-week time periods. Major home run. I mean, if you like baseball metaphors, this is stepping up to plate, not knowing if you can hit the ball, and hitting one over the green monster in Fenway. I mean, huge milestone. The unfortunate thing is that the time to which the patients progressed on this drug wasn't that long. It was only about 7 to 9 months. And we've subsequently spent the last several years of research trying to pick apart exactly why these tumors become drug resistant. Now, it's not everybody. We have patients that have had all of their tumor go away with this medicine, and they are off medicine and cancer-free, near as we can tell. But they're the minority, so we need to figure out exactly how we can stretch that mile with the drug. I'm going to move briefly over to CTLA-4, and Dr. Annabaca described this a few moments ago. This is James Allison. He really is the father of the field in terms of a description of CTLA-4 as a potential strategy to treat cancer. And this also was back in the 90s, so the same era that Brian Drucker was working, Jim Allison was in a lab at UC Berkeley doing the same kind of things with immune therapy. Those lab discoveries led to an FDA approval of ipilimumab in 2011, and it really paved the way for all of the work that we're enjoying today. What was so profound about this is it was a totally different way of thinking about our immune systems. What he described was basically taking the brakes off of these T cells, which really are the Marines of your immune system. They're out there, they're trying to kill cancer. Well, we want to take their brakes away, and that's what Jim Allison really described, was by taking that brake system off, we could get cancers to respond. I tell you that story because it takes many, many steps from a lab discovery like that to make an impact in patients. And this is a trial. This is an example of a phase 3 trial that led to a drug approval of ipilimumab in adjuvant melanoma. This is the area where I work. Well, adjuvant therapy, for those of you who don't know, it's like an insurance policy. Let's say you've had a good surgery. All the cancer's out, but you need an insurance policy to be sure that this cancer is not going to come back. Well, this was the trial that established that. I think what's unique is that it did require a placebo, and that placebo control allowed us to demonstrate that it did indeed delay the time to which cancer comes back by about 1 year. It was a pretty big advance, and it led to an FDA approval of a new drug. What we care the most about, obviously, is are these people living longer? And we will learn that, I'm hopeful, in a few short weeks in Copenhagen, where the longer-term data of this entire population will be presented. From CTLA-4, we go to PD-1, and drugs like nivolumab and pembrolizumab, with Dr. Kujakar will describe in great detail, really were developed by a team of scientists, one of them being Tasuku Honjo, an investigator in Kyoto, Japan. What he's done with his career is very similar to Sherry. He's developed tons of knockout mouse animals, some of which I actually worked with in the lab when I was doing that kind of thing. Unbelievable impact in an entire institute of tumor immunology, all focused on mouse modeling. Well, his discovery of PD-1 in that laboratory led to these two, Arlene Sharp and Gordon Freeman, investigating it in cancer cell lines. And then finally, Lei-Ping Chen really pushed this over the finish line and showed, wow, we can inhibit that, and boy, it can really work in cancer. In 2014, they shared the distinguished William Coley Award for research in tumor immunology. And it led quickly to multiple research studies in melanoma. This one led by Tony Rebus, led eventually to the FDA approval of pembrolizumab. Its name has actually changed since the original article. This is Omid Hamid, whose name is shown here. This work, led by Michael Postow, was one of probably 3 trials that were necessary to get this over the finish line. And Dr. Kujakar, while she was at the Moffitt, was one of the first investigators working with these PD-1 molecules as they made their way from phase 1 to phase 2 to phase 3. Well, why is this important? So now we're talking about swimmer plots, which you were introduced to just a little while ago. And these are patients who have been treated with pembrolizumab and followed forward in time. These arrows are those that are continuing to have a response. These blue, or rather yellow triangles, are the time at which it takes to measure a response. And probably the most important thing are these red triangles. And those are the people that see every last spot of cancer in their body disappear, and we call that a complete response. Well, it's drugs like this, particularly that harness the immune system, that can get that deep, complete response, are the ones that I think are going to drive us to potentially curing melanoma. The other great thing about these drugs is that they are incredibly safe. Whereas other types of compounds that we've worked with cause a lot of illness, only about 12% of these patients will experiencing something bad enough that they might see a doctor for. Maybe it's a cough, maybe it's diarrhea. Oftentimes these side effects are in the category of bothersome, but not so bad that we can't control it. And that leads to this phase 3 trial. So again, we have to prove that these drugs work better than their standard, and that is the work that we're doing in adjuvant melanoma, and we're comparing that to the standards interferon and now ipilimumab, which has been added to this trial. This is what the design looks like, and it's simply a group of patients here who have had good surgery, who will then go on to get either one of these therapies, and they have a choice between interferon or ipilimumab in the standard of care arm. It takes a lot of sites to do this, and so far we have opened over 135 sites nationwide. There's several here in Utah. Both the Huntsman and the Intermountain healthcare system have this trial open. Atlanta has it open. Thank you, Dr. Kruchikar. No, I missed. I'm going to fix that spot. I know you're registering because I've seen your numbers. So moving right along. No, it really does take a lot of people contributing to these things to make them a success. And it's through this work that I think that we're going to take the bar from where it sits right here to hopefully right here by the time I get to retire. And I want to retire early, so we better make it happen fast. The final thoughts that I want to share with you before we take a break is that I think from a patient point of view, decisions to participate in research are not so easy, right? You're going to be signing yourself up for something that may or may not work. And I think what you have to ask yourself, if you ever find yourself in the position, is really understanding, how is this new drug supposed to be better than what the standard is? Is this something I really want to do? Is this a randomized study? Do I have a chance of getting some other treatment that maybe I don't necessarily want? Is there a placebo? Does that really make sense to me? Am I comfortable with that, given my certain circumstances? What additional testing am I going to have? Do I have to have a blood draw every day for a week while I'm participating in this trial? Do I have to have my tumor sampled over and over again? Obviously, these things are really important to get comfortable with, and if you're not comfortable, that is okay. Those of us that screen our patients for clinical research never feel bad when they say no, because if it's not for you, it's not for you, and that's the right answer. What are the side effects of that drug? What are the other options available to me when I'm facing this challenge? And then finally, what's the schedule? And I think for those of us that live in the Mountain States area, really have to think of this importantly. Some of our patients driving 10 hours a time to get here. Obviously, these clinical research protocols that require weekly visits start to ask a lot of them. I'm always grateful to the person who's willing to do that. And I've had a number of in my clinic over the years who've done it from the nether reaches of Montana, and they really will. They'll drive those 10 hours to come and see us and try and push this bar forward to help clear melanoma. The study factors also are important to consider, and some of you may or may not have been through a process where you didn't enter a trial because you didn't have the right features. That's hard, but we have to be careful about picking the right cancers, the right stage, the right tumor locations, whether or not previous treatment could influence the outcome of this next trial, whether or not there's tumor in the brain. I'm going to pause here for a moment because it probably is one of the hardest areas in melanoma to work with. A lot of the studies will exclude brain tumor involvement, and it has a lot to do with the fact that it's harder to predict the future of people that have had that unfortunate complication, and the studies will exclude them. And it's been our effort over the years when we work with our industry colleagues to say, wait a minute, we need more trials for brain mets. And we're in the process of opening one now here at the Huntsman. I'm sure that Emory has several, and it really remains our next frontier in melanoma is to tackle this in a very sophisticated way. How do you find out about trials? Well, your doctor is probably the best choice for starters, and they should know where these are available at their institution and perhaps even around the country. And from my point of view, I see the whole US as our clinic. If I don't have a trial, I will send my patients to MD Anderson, or I'll send them to Emory. You really need to think that broadly when you're considering clinical research. We have on our website, and I've listed the URL here, a terrific trial selection algorithm that will let you go by cancer, right on down to the specific study. The US government also has a terrific site that does something similar on a national level. And then finally, the AIM at Melanoma Foundation, Sam has provided some terrific materials, and what you've received is a handout. And through them, they can identify physicians and trials and options. They're a tremendous research resource. Well, what does this look like? I took the NCI's webpage as I was preparing this talk, and I plugged in melanoma. I plugged in our zip code, which is 84109, real close to our cancer center. And then I plugged in age 18, because I really do want to be 18 again. I'm getting older. But it came up with several studies, and you can see that we're listing some here. This is that stage 3 trial I talked to you about for adjuvant melanoma. Dr. Annabaca's study is listed here in terms of the efficacy of TVEC. And there are multiple other studies that will just pop right up for you if you want to go through this exercise to see what sort of resources are available. I'll end my talk with a brief note about prevention. So these are my great-grandparents. Her name was Grandma Sis, and his name was Doc Grossmann. He was a realtor, but they called him Doc. And what they are sporting here was what sunscreen looked like at the turn of the century. I think they were honestly ahead of their time. We've really come to think more and more that covering up our skin is probably even better than some of the sunscreens. I think sunscreens are great. You can't replace them. You got to put them on, and you got to put them on about every hour or two when you're out in the sun. But good clothing really makes a difference. And I'll just finish with a final thank you to the AIM at Melanoma Foundation. Val Guild has actually been, personally for me, a tremendous advocate for my career and for the work we do here at the Huntsman. And I'm so glad to have Sam here today hosting with all of us. Thanks.